new study, scientists have reported mice living for up to 58 months. Now, that might not sound all that impressive until you realize that the typical lifespan of a laboratory mouse is only about 24 to 28 months. In other words, some of these mice lived more than twice as long as normal. And the most surprising part, the effect didn't come from a drug, from gene therapy, or some kind of stem cell treatment. It came from something the researchers are calling telomere rivers. A recent study led by Professor Alessio Lana of Oxford University has reported something that at first glance seems almost unbelievable. Researchers are suggesting that immune cells may be able to release particles that carry telomeric DNA, the protective DNA sequences found at the end of chromosomes. Now, according to the study, these particles can enter the bloodstream and circulate throughout the body. The researchers call these particles rivers of telomeres. And in experiments with mice, the study reports something even more surprising. When these particles were transferred into older animals, the researchers observed signs of tissue rejuvenation and even reported a significant increase in lifespan. Now, if these findings hold up, they could represent a completely new mechanism related to aging. But before we get into that, there's this. If you're fascinated by longevity science and want to learn how to reduce your biological age and extend your health span, then why not join us in Longevity Lab, a free community on school.com. This space was created for like-minded people with an active forum of hundreds of members from all over the world where you can connect, hang out, learn, and share. There's also a classroom tab with free online courses and coaching cohorts and a members map to see where in the world your new friends live. On School, there's none of that typical social media rubbish. It's exactly how social media should be. The link to join is on screen, it's in the description below, and it's in the pinned comment. We'll see you there. Okay, before we look at what this study is all about, we first need to take a look at telomeres. Telomeres are protective, non-coding DNA sequences located at the ends of our chromosomes. You can think of them like plastic tips on shoelaces. Their job is to protect the genetic material in our chromosomes from damage during cell division. Now, here's the problem. Every time a cell divides, the telomeres become slightly shorter. Over time, as telomeres continue to shorten, cells eventually reach a point where they can no longer divide normally without destroying valuable genetic information. Now, at that stage, many cells die. They enter apoptosis, programmed cell death. Other cells enter a state known as cellular senescence, where they stop dividing but remain metabolically active. Because of this, telomere shortening has long been considered one of the biological processes that contributes to aging. That's why scientists studying longevity are very interested in understanding how telomeres are maintained and whether it might be possible to slow, stop, or even reverse their shortening. And the main avenue of investigation has been telomerase, an enzyme that can actually lengthen telomeres. But this study raises a much stranger possibility. What if telomeres could actually move between cells? To understand what the researchers observed, we need to look briefly at how certain immune cells interact. One of the key players in this study is a type of immune cell called CD4 helper T-cell. CD4 T-cells are sometimes described as the coordinators of the immune system. They help organize immune responses by communicating with other immune cells and directing how the body responds to infections. Another important immune cell is the antigen-presenting cell, often shortened to APC. Antigen-presenting cells act like messengers. They capture pieces of pathogens, such as viruses or bacteria, and present them to T cells so the immune system can recognize that as a threat. When an APC and a CD4 T cell interact, they form a very tight and specialized connection known as an immune synapse. This junction allows the two cells to exchange signals and coordinate the immune response. And it was during this interaction between APCs and CD4 T cells that researchers began noticing something unusual. During experiments involving antigen-presenting cells and CD4 T cells, they detected telomeric DNA fragments appearing outside of the cells. At first, these fragments looked like isolated pieces of DNA. But as the researchers examined the samples more closely, they saw that the telomeric material was not randomly distributed. Instead, the DNA appeared organized into elongated extracellular structures that extended throughout the surrounding fluid. Now, under the microscope, these structures looked almost like branching networks or streams of genetic material moving through the extracellular space, the space outside of the cells. Because of this unusual appearance, the researchers began referring to them as telomere rivers. Further analysis suggested that these rivers were composed of extracellular particles containing telomeric DNA. The researchers were also able to detect similar particles circulating in blood plasma, indicating that these structures were not limited to a laboratory dish, but could also exist in the bloodstream. Based on their observations, the team concluded that these particles likely originate during the interaction between antigen-presenting cells and CD4 T cells. In other words, when certain immune cells communicate with each other, they may generate particles containing telomeric DNA and release them into circulation. Now, this raised an obvious question. What exactly are these telomere rivers and how are they actually formed? So, to answer that question, the researchers proposed a step-by-step mechanism for how telomere rivers might actually form. The process begins at the immune synapse, the specialized contact point where antigen-presenting cells interact with CD4 T cells. Previous research had already shown that antigen-presenting cells can transfer telomeric DNA fragments to CD4 T cells during this interaction. This transfer can help extend the telomeres of the T cells themselves, potentially improving their longevity and function. But in this new study, the researchers noticed something additional happening during the exchange. Not all of the transferred telomeric DNA appears to remain inside the T cells. Instead, some of this material may become packaged into extracellular vesicles. These vesicles are small, membrane-bound particles that cells often use to transport molecules between tissues. According to the model proposed in the study, CD4 T cells incorporate some of this donated telomeric DNA into their own chromosomes, while excess telomeric material is packaged into vesicles and released from the cell. These vesicles then enter the bloodstream and circulate throughout the body. The researchers refer to these circulating vesicles collectively as telomere rivers. Analysis of the particles suggests that they contain not only telomeric DNA, but also a variety of proteins associated with cellular signaling and stem cell-related pathways. At the same time, the particles appear to lack certain metabolic enzymes that are commonly found in other types of extracellular vesicles. The researchers speculate that this unique composition could allow the particles to act as biological signals that influence age-relating processes in distant tissues. But a hypothesis is only meaningful if it produces measurable biological effects, right? So the researchers designed experiments to test whether these particles could actually influence aging in living animals. To test the biological impact of telomere rivers, the researchers conducted a series of experiments in mice. First, they isolated telomere river particles from the blood of young mice, where these structures appear to be present at the higher levels. They then transferred these particles into older mice to see whether they would affect age-related biology. Over time, the researchers began to observe several changes in the treated animals. In multiple tissues, the study reported reductions in cellular senescence markers, including markers that are commonly associated with aging cells. The researchers also observed decreases in certain inflammatory signals, which are often elevated during aging and are believed to contribute to age-related tissue damage. At the same time, the study reported increases in telomere length in several different organs. And these changes were not confined to a single tissue. The researchers detected effects in a variety of organs, including brain, liver, lungs, kidneys, and heart. Taken together, these findings suggest that the particles may influence biological processes related to aging across multiple organ systems. In other words, rather than acting locally in one tissue, these circulating particles might be capable of sending signals throughout the entire body. Now, that result alone would be pretty remarkable. But the most surprising finding in the study did not involve cellular markers or tissue changes. It involved lifespans. To test whether telomere rivers might influence lifespans, the researchers performed a separate experiment. Now, in this study, rivers were collected from animals in which the immune system had been stimulated to produce these particles. The researchers then transferred those rivers into older mice. The animals were then monitored over time to see whether the treatment would influence survival. According to the results reported in the paper, the mice that received the telomere rivers lived substantially longer than untreated animals. The study reports that the median lifespan for these elderly mice increased by roughly 17 months. Now, for mice, that represents a very large shift in lifespan, on the order of roughly 60% or more. But what was even more striking is that some of the animals lived even longer. The maximum lifespan achieved was 58 months. For context, the typical median lifespan of a laboratory mouse is around 24 to 28 months, meaning that some of these mice lived more than twice as long as a typical mouse. Now, it goes without saying that results of this magnitude are highly unusual in longevity research, which naturally raises an important question. How seriously should we take these findings? As intriguing as these findings are, it's important to place them in the proper scientific context. First, this study is currently a preprint, which means that it's been released publicly, but has not yet undergone peer review. Peer review is a process where independent scientists evaluate a study's methods, results, and conclusions before it's formally published in a scientific journal. That process often leads to revisions, additional analysis, or sometimes challenges to the interpretation of the results, although I'm not sure how else you could interpret a 60% increase in lifespan. Second, the mechanism proposed in this study is highly unusual. While extracellular vesicles are well known in biology, the idea that immune cells might release circulating particles containing telomeric DNA that influence aging across the body is a completely new concept. Discoveries that propose entirely new biological mechanisms always require independent replication before the scientific community can determine whether the findings are robust. That means other research groups will need to reproduce these experiments and confirm the results. Until that happens, the findings should be viewed as an intriguing but very early stage discovery. So, what should we take away from this study? The researchers are proposing that immune cells may release particles containing telomeric DNA that travel through the bloodstream and influence age-related processes in other tissues. They refer to these particles as telomere rivers. In mice, the study reports signs of tissue rejuvenation and a substantial extension of lifespan after these particles were transferred into older animals. If future studies confirm these findings, it could represent an entirely new direction in longevity research. But for now, this idea remains as a fascinating hypothesis that scientists will need to test much more carefully. But it's pretty damned exciting. Okay, that's all I got. Don't forget to check out the Longevity Lab. The link's right here. That's it for me. I'm out of here. Catch you guys next time.